Hi, in this video we're going to talk about some extensions to Mendel's theories of inheritance. Now, when we talked about Mendel's experiments, we always talked about characters that had alleles which showed complete dominance. Now, if you recall the example of the monohybrid cross, where we looked at cross between purple flowers and white flowers, we saw that the allele for purple color was dominant to the allele for white color. And so that the heterozygous organism that had an allele for purple and an allele for white only appeared purple, because the effect of that dominant allele completely masked the effect of the recessive one. Now, some alleles actually, it turns out, show incomplete dominance. And in these, we have a heterozygous phenotype that is distinct from the two homozygous phenotypes, and generally looks like a blend between the two, or is sort of like an intermediate between the two. So, if we take a look at this example of a cross between this red flower and this white flower, we see that the red flower is homozygous with these two capital R alleles. The white flower is homozygous with these two lowercase r alleles, and their offspring are all heterozygous. But those offspring neither appear red nor white, but actually show up pink, because both alleles are, because they're heterozygous, they have both alleles, and their heterozygous phenotype is distinct and a blend, in fact, between the two colors of the homozygous plants. So, this is an example of incomplete dominance. However, we also see something called codominance, and this is a little different. Now, thinking about that flower example we just talked about, if those red and white alleles had shown codominance instead of incomplete dominance, our heterozygous organism would look like this. Rather than showing a pink color, a blend between the white and the red, it would show both white and red, but separate from each other. So in codominance, the effect of both alleles is seen in the phenotype, but is distinct. So, this is actually how blood type inheritance works. Now, there are three alleles we're going to look at with blood type. There is the allele for type A blood, which codes for a particular carbohydrate that comes off the surface of those red blood cells, and that is type A. Then there is type B blood, and an allele for type B blood, and that individuals with type B blood have a different type of carbohydrate projecting from the surface of their red blood cells. So there's also a recessive allele with blood type that we symbolize with the lowercase letter I. And if we were to cross these two individuals, we'd see the outcome for the offspring. We could have offspring with type AB blood. That would be the example of codominance, right? We have both A and B carbohydrates on the surface of those blood cells, and we see the effect of both alleles. They are codominant. One does not mask the other. However, individuals that have B and the recessive allele will only express those B surface carbohydrates, and individuals with A and the recessive allele likewise will only have the A carbohydrates, and individuals with two recessive alleles, it turns out, don't have anything on their surface. They don't have these doodads or these doodads. They have nothing there, and those individuals are said to have type O blood. So type O blood is the homozygous recessive allele combination, whereas AB is both of the codominant alleles together. Alright, let's flip the page and look at some other extensions to Mendelian inheritance.